In this session, I'll discuss about mapping of bacterial genome. In bacteria, the genes are mapped by means of interrupted mating experiments because the bacteria does not undergo meiosis. The mode of replication in bacteria are by three ways: conjugation, transduction, or transformation. In case of bacteria, transfer of DNA takes place from HFR cells to F- cells. Chromosome transfer occurs within the integrated F factor and proceeds in a continuous direction. The time required for individual genes to be transferred is related to the relative position on the chromosome. Jacob and Wohlmann in 1957 were the first to develop the technique of interrupted mating to map the genes in bacteria. They used HFR Haynes strain as a donor. These strains were sensitive to antibiotic streptomycin, resistant to sodium azide and infection by bacteriophage T1, prototropic for threonine and leucine, and able to break down lactose and galactose. The F1 recipient cells used were resistant to streptomycin, sensitive to sodium azide, infection by bacteriophage T1, autotrophic for threonine and leucine, and unable to break down lactose and galactose. The two strains were mixed in the nutrient medium and allowed to conjugate. At different time intervals, the mating mixture was diluted and agitated using blender to separate the mating cells. These were then plated on a selective media containing streptomycin and lacked leucine and threonine. The HFR donor cells and the recipient F cells would not grow in this media as HFR cells are sensitive to streptomycin and recipient F cells are autotropic for leucine and threonine. Only the cells that underwent conjugation and received the leucine and threonine would survive in the selective media. These cells were then tested for other genes that might have transferred from the HFR strain. The percentage of streptomycin resistant leucine and threonine plus cells were received specific alleles were plotted against the duration of conjugation. It was observed that a latent period of 8 minutes was required for the conjugating cells to make contact. And then the genes from the bacteria of HFR is transferred in an oriented linear manner to the F- strain. The first donor gene appeared to be was sodium azide gene at 9 minutes, followed by TON resistant gene at 10 minutes, then LAC at 18 minutes, and so on. In bacterial mapping, construction by interrupted conjugation, the base unit of distance between the genes is minute rather than frequency recombination percentage. The figure here shows the graph plotted against the percentage of streptomycin-resistant, leucine-positive, and threonine cells received specific alleles against the duration of time of conjugation. These are certain biochemical markers used in genetic analysis of yeast genome. Advantages of linkage maps. It allows to understand the overall complexity and genetic organization of a particular species. It helps to study the evolutionary relationship among different species. In case of humans, it can be used to diagnose and treat inherited human diseases. It helps to predict the likelihood of a couple to produce a certain inherited disease. It provides an overall information in improving agriculture important strains through selective breeding programs. The limitations of linkage maps are recombination frequencies are always underestimated in genetic distances for larger distances due to interference. Crossing over is inhibited in some regions while there are certain hotspots for recombination in the genome. In humans, the frequency of recombination of loci of most chromosomes is higher in female than in males. Thank you.